Okay, so welcome everyone. My name is Bernhofer, and today I'm giving a talk about TMSEG, which is a transmembrane protein and helix prediction method. But first, a short excursion to signal peptides, because I heard you haven't heard about them yet, but they will be important later. So basically, what are signal peptides? Signal peptides are just short peptides at the N-terminus of secreted or other proteins, though there are some exceptions where the signal peptide might not be at the N-terminus, but somewhere along the sequence or near the end. What is a signal peptide? Well, basically you could call it a zip code for the protein transport, so it signals the transport proteins where a protein should end up, if it should end up outside of the cell or in a different cell organelle. So that's what signal peptides are for, and that's where the name is coming from. Typically, a signal peptide is cleaved after the protein is at its final destination, but some, like signal anchors, which are signal peptides that are then embedded in the membrane, might still be on the protein and are not cleaved. And the other thing which makes signal peptides that important for transmembrane proteins and their prediction is that signal peptides share multiple features with transmembrane helices. So for one, the length might be similar. Signal peptides can be up to 30 amino acids, but they can also be much shorter than transmembrane helices, so they might only be 6 residues long or maybe 10. But they have a hydrophobic core region, which is the green stretch. And this can be between maybe 6 and 15 residues, so it can easily be confused for transmembrane helix, which is also hydrophobic. And many signal peptides, unfortunately, also have a helical structure, so they are even more similar to transmembrane helices. The other thing is that the N-terminal part of a signal peptide often is positively charged, which once again is a signal that we want to use for transmembrane helix prediction as the positive inside rule, which I think you heard about last lecture, helps us to determine which soluble loops of a transmembrane protein are on the cytosolic or on, for example, the extracellular side of the membrane. So these two features, the hydrophobic region and the charged region, cause quite a confusion for prediction methods between signal peptides and transmembrane helices. So this just short explanation why signal peptides are important for this prediction and what they even are. So now, last time you heard about transmembrane proteins and their structure prediction. You had a short evaluation of methods, and now you're hearing about TMSEG, which is a method developed by me, and Edda Kloppmann was part of the team, and also Jonas Reeb helped. And here you can see basically the performance of TMSEG compared to other state-of-the-art methods like Polyphobius, MEMSAT3 and MEMSAT SVM. An older method from Rostlab, PhD HTM, and a simple baseline predictor. And while you can see a trend on the prediction performance in the figure, you can also see that the error bars are so high that we can't really say that one performs significantly better than the other, because factoring in the error, maybe we can distinguish PhD HTM and the baseline predictor, but for example, even here, Polyphobius and PhD HTM are within one standard error. So this is the paper for TMSEG, which was published in Proteins, if you want to look it up and get a more detailed view on TMSEG and the methods behind it. Okay, so the question is, why do we need yet another transmembrane prediction method? Well, one thing is now we have more data available than we had a few years ago. And many of the methods I just showed you, like Polyphobius and MEMSAT, are already a few years old. PhD HTM is, I think, 20 or 25 years old. So in the meantime, we have more proteins we can train on. So hopefully we get better prediction performances. Another way would be to just retrain the old methods, but unfortunately most of the people just don't do it. They have their old published methods and don't use the new data. Some might publish new methods based on the old ones, but that's not that often. Also nowadays machine learning algorithms got faster. They were more optimized for the training time. We also have more computing power, so we can just handle much more data and more complex prediction algorithms. And the other thing is related to this is just runtime. Many of the older methods, for example MEMSAT SVM, take several minutes just for one protein. If you, for example, look at the human proteome of 20,000 proteins, then you can just do the math. If you take 5 minutes per protein, how long you will take for the whole proteome. Okay, so to start a prediction method, like you already know maybe from the exercise, you need a data set. In this case, we want to predict transmembrane proteins, so I need first my positive samples, so my transmembrane proteins, which I got from OPM and PDBTM. Those are protein 3D structure databases which are explicitly for transmembrane proteins. They take the structures from PDBTM, then run an algorithm they developed over the 3D structures to determine where the membrane bound should be. And then there's also a level of manual curation, so it's not just put the algorithm on it and then let it sit, but they also look at the structures.
So I assume you meant they take the structure from PDB?
Yes.
From PDBTM. So
oh, sorry, yeah.
Why do you not see, why do you need to determine where the membrane is? Why don't you see that in the structure?
That's in during the structure. So when you crystallize a protein, then oftentimes it's just not in the membrane, because the membrane you can crystallize. That's also a reason why transmembrane proteins are hard to determine the structure, and you need special detergents, for example, if you want to simulate the membrane. And in the radio diagram, then you can get from those crystals, you don't see the membrane anymore. Okay. But the bad thing is that as you can see the membrane in the structures, and the algorithms, well, they use two different algorithms for OPM and PDBTM, so they don't always produce the same results. And when gathering this data set, I noticed that sometimes the annotations for where is the membrane boundary, so where are the transmembrane helices, might be off a few residues, but sometimes one database annotates 3 transmembrane helices and the other one annotates 4. So there are sometimes even bigger differences. Then the next step was to, well, map those PDB structures to UniProt sequences, because PDB structures don't always have the whole sequence. They might cut out some loops which are hard to crystallize, and I wanted to train on the whole sequences. So for this I used SIFT, which is basically a database of different ID mappings. So they have the PDB ID, and you can map it to UniProt. You can map it, like I did, then to OPM PDBTM, which also oftentimes use the PDB ID. And for, I think, 6 or 7 of the most popular databases, they have those ID mappings. And the last thing was that I have to reduce the redundancy, because when I want to make a prediction method, I have to watch out for similar proteins or even identical sequences in the training and the test data set. For this I've used UniProt at an H-value cutoff of 0. Is everybody familiar what H-value of 0 means? Should I explain it for someone? Yeah, way around. Okay, so if you remember the HSSP curve with the midnight zone. Okay, so basically the curve that you've seen in the graph is, okay, I can just sketch it really quick. Okay, so here We have the percentage identity of the alignment. Here we have the length of the alignment, then HSP curve is somewhere around there. Okay, so and typically the HSP curve that is shown corresponds to an H value of 0. So everything above the line has an H value of higher than 0. So we could infer that they have the same structure. Everything below is in the midnight zone where we just don't know. So they might still have the same structure, but we can't say for sure. And so basically what I have done with UniQprot is that I only have protein pairs in my dataset which are below this curve. Okay, so I've extracted roughly 1,000 proteins from OPM and PDBTM, and in the end I was only left with 166 transmembrane proteins, which just comes from the fact that many of the structures are for the same protein. So I have one sequence maybe 10 times, and then I have some related which have 90% sequence identity, and all of them got thrown out. The other thing is I don't just want to predict where are the transmembrane helices or is it a transmembrane protein. I also want to predict the inside-outside topology. This annotation I've got from OPM because at the time when I created this dataset, PDBTM didn't have this information. Nowadays you can also look up the inside-outside topology. But when I created TMSEG, I had to take the annotation from OPM and also then transfer it to PDBTM, which once again caused some problems because if the annotation for transmembrane helices are not always matching, then I sometimes also got problems with the inside-outside topology, and I had to look at the protein structure and to my best knowledge try to determine which side is inside and which side is outside. So now that I have my transmembrane proteins, so my positive samples for the prediction, I also need a negative dataset. For this, I've taken the dataset that was used to create signal P, which is a signal peptide predictor. It has soluble and membrane proteins, and it has, most importantly, proteins with and without signal peptides, the ones we talked about earlier. And first thing was, I only have 166 transmembrane proteins, so I want to keep them all. And to do that, I first threw out all proteins that are similar to my 166 transmembrane proteins in the signal P dataset. And then I've also reduced the remaining proteins in the signal P dataset with an H value of 0 to have complete non-redundancy between the datasets and in the whole dataset basically. And what I was left with was roughly 1,100 soluble proteins and 300 membrane proteins. But those membrane proteins from signal P I did not use for the later evaluation and for training of some of the machine learning tools later on, but I will mention that when we get there. But I did use them for the initial classification in membrane protein or non-membrane protein. The reason for this is simply because while the signal P dataset was annotated for experimentally defined transmembrane proteins, the position of the transmembrane helices were not. So I could trust that it was a membrane protein, but I couldn't trust where the transmembrane helix was annotated. Okay, now I have my two datasets, my positive and negative dataset, and what I did was divide it into 4 subsets. One of those 4 subsets I called the blind test dataset, which is this split of 41 transmembrane proteins and 285 soluble proteins from the signal P dataset. The remaining 75% I also split into 3 sets for 3-fold cross-validation. I think you should be familiar with cross-validation from the exercise and the last lectures. So, or should I go over it again, what this means? Okay. And well, then just at the end I combined the corresponding splits into one data set. So I combined the first split from the transmembrane proteins with the first split from the soluble proteins into one data set. And this was then basically my first split for the cross validation, second and third one. Okay, so before we get into what types of machine learning tools I've used for TMSEG and how I've used them, I want to give you a short introduction to classification trees, because while I think everybody should be familiar with neural networks, and correct me if I'm wrong on this one, okay, I guess not all of you know how a classification tree works, or in my case, later on what I've used, a random forest. But basically a classification trees, it gets a set of samples, so the training samples, and it gets a set of features for each of these samples. And then at each node in the tree, it picks one of the features and tries to determine the best feature and the best threshold for this feature to divide the samples into the separate classes. What I mean by this is on the next slide. So in this case, it's an example for a classification tree, and these are my samples and my two features x1 and x2. And in the first step, the classification tree sees, okay, x2 is probably the best for the first split, and decides to use a threshold of 0.7, which more or less divides the data set into the ones and then 2 and 3. For the subsequent splits, it decides to take feature x1 with two different splits, because everything that is above 0.7 has to be differently divided than everything that is below this threshold. And based on this second split, it can divide most of the classes into 2 and 3, or into 1 and 2. And the final output of this tree wouldn't just be a, well, binary, or in this case an absolute prediction. So for example, for this leaf node for 3, it wouldn't just say, okay, 3 with a probability of 100%, but 3 with a probability of, I don't know, maybe 90, or basically based on the distribution of classes in this sector. So?
I would say it's 85% because you also have the ones there and not only the twos. So
yeah, it was
even be 60.
I don't know, I didn't count it. But it's basically based on the distribution in this sector of the classes.
You have like 20, 23s and around 10 other numbers. So yeah, it's like a 2/3, 1/3 split in the down right quadrant.
2, 4. No, it's 7 other classes in this one. 3 ones and 4 twos.
There's one at the topmost 3.
Ah, sorry. Okay, yeah, it's 8. Okay. But, well, I guess you get the idea. So basically the probability that I get for the prediction is based on the distribution in this final sector. So next step, instead of one tree for a random forest, we're using, well, a forest, so multiple trees. And for a random forest, you most of the time have 3 parameters. One is the size of the forest, so how many trees do you use? Second one is how many random features are you using for each split. So instead of looking at the whole feature space at every node to split the data set, you randomly select a subset, and then from this subset you pick the best feature. Because if you wouldn't sample from the features, then every tree would look the same, because every tree has the same features, so it would always make the same splits. And by randomly sampling the features, you get a different distribution and different split for every tree. The third parameter that you can use, but don't always have to, is to also randomly sample from your samples that you put into. So instead of using the whole training set, you could only use 90% or 80% of the data set. This further introduces a randomness which can help to make the tree or the random forest more robust against outliers.
So I guess there's a typo, right? This was supposed to be N1.
Oh, yeah, sorry.
Small n.
Yeah, or n prime, something like that. Just to distinguish it, yeah. Okay, so do you get an idea how a random forest works? Well, can you imagine how it works as a machine learning tool? Okay, so one question. Now we have this random forest. We put a sample into it, let the random forest do a prediction. How do you think we come to the final prediction of the random forest as a whole? I mean, we have multiple trees, but how do we get a final prediction? Does anybody have an idea? Okay, well, it's actually quite simple. It's basically for every tree you get a probability for one of the, so you get a probability for the most likely class, and then you just build the average over all predictions from all the trees, and whatever class has the highest probability based on this average is then your final prediction of the random forest. Okay. Yeah, so random forests, why did I explain them? Because they weren't that popular in the beginning. As you can see, neural networks have been popular since almost 40 years now. But random forests only became popular in recent years. And I don't have an up-to-date statistics for 2017, but I could imagine that they're much higher now because random forests perform well in many different fields. Okay, now that we have it down how a random forest works, well, how did I use a random forest for TMSEG? TMSEG basically is divided into 4 different steps, and for the first step I have one random forest, which I'm using for a per residue prediction. I'm using a sliding window approach, which you should be familiar with. So basically I have my window of 19 residues, and the prediction that I'm making is for the central residues of those 19s. For the parameters, my random forests it uses 100 trees and randomly selects 9 of the features. On the next slide I will show you how many features we have. And I didn't also sample from the training set, but I used 100% of the training set. The output from the random forest is a probability for each of my 3 classes. I divided the residues into signal peptide residues, transmembrane helix residues, and the rest, which I called soluble residues. And to make the internal calculations a little bit faster, I wanted to use integer instead of doubles. That's why I multiplied it with 1000 and then just used integer. So the next slides, when we're talking about scores, it's basically the probability that I'm getting from the random forest multiplied by 1000. So a score of 1000 means the output was 1.0, so 100%. Score of 500 means 50% probability. So if we run a protein through this random forest, we might get something similar to this. So on the y-axis we have the score. Like I said, 1000 correlates to 100% probability, 550 and so on. And on the x-axis we have the position or the residue within the protein sequence, and we have our 3 classes from the output for signal peptide, non-transmembrane and transmembrane. So if we just look at this graph, we might suspect, okay, here we have a signal peptide. Then we might have a transmembrane helix here. This looks more like noise, and here I have a surprisingly long transmembrane helix of something like 60 residues, which is most likely too long. But we'll come to that later. Okay, what features did we use? We use two global features. First one is the global amino acid composition of the protein. Second is the protein length, although we didn't use the absolute value of the protein length, but we binned it. So for example, a 0 corresponds to a length of up to 120, a 1 to 121 to 240, and so on. As local features, so for the window of 19 residues, we use the PSSM score. Yes?
How did you select the window of 19 residues?
Okay, so how did I select the window of 19 residues? I first intuitively thought, okay, most membranes span something around maybe 17 to 21 residues. So I started in this ballpark, I started with 19. And then during the cross-validation, I also tried to optimize the parameter, but in the end, 19 turned out to be the best one. Okay, the other thing besides the PSSM scores, so basically for every position I have the 20 PSSM scores from a PSSM matrix. Have you all heard what a PSSM is from Cyblast? Okay. Second feature, or two features, is the distance to the N and C terminus. Once again, it's binned, so it's not the absolute value, but just within the range. And then we have four physicochemical features for the residues, but in this case I wanted to be more local to not get too much noise from the neighboring residues, and I used a window size of only 9. And this is the average hydrophobicity of the residues, because we all know transmembrane helices mostly consist of hydrophobic residues. I also have the percentage of hydrophobic residues. I have the percentage of positively and negatively charged residues, and I have the percentage of polar residues. Okay, so like I said, I've used the PSSM score, which for this random forest and the window approach is quite intuitive. So for every position, I just have 20 input features. So for a window size of 19, I would have 19 times 20 input features. But the other thing is, when I wanted to use, for example, for the average hydrophobicity or for the amino acid composition, I didn't just want to use the original sequence, I also wanted to include this homology information from the PSSM. So I had to think of a way how could I integrate it. And the way that I used is, I looked at my window. In this case, we have an example just for the first 5 residues. That's, let's say, is our window. How many positions do I have with a positive PSSM score? So where are which residues are more likely than just by chance to appear in the sequence? And how many positions do I have with a negative PSSM score? So which are less likely to appear? And with this method, I could then establish, for example, an amino acid composition. So if I want to have the the positive amino acid composition for M in this window, I look at the window, I look at the M column, how many positive scores do I have? In this case, I have only 1. I had 16 positive scores in total in this window. So I say my positive amino acid composition for M is 1/16. The opposite is the negative amino acid composition. In this case, I have 3 negative scores. I have 79 in total. So once again, I have 3/79 of a negative amino acid composition for M. And that way I calculated it for all of the 20 amino acids, and for example also for the percentage of positive charged or hydrophobic and so on. So in this case, for positive charge, I look at the columns of positive charged residues. How many positive scores do I have in there? In this case I have 2, so it's 2 divided by 16. Yes?
Yeah, I have a question. So while I do understand what it means biologically, if the PSSM score is like negative or positive, I don't quite understand how these fractions translate to a meaning in a sense of a prediction.
Well, it's not a prediction, it's a feature for the prediction method. But
All right.
okay. No. But the sense behind it is, if I want to make the amino acid composition just for the sequence, then well, I have the sequence, but there I am losing the homology information from the alignments from BLAST. But in this case, I can see, okay, in a different, so let's pick one that has more positive ones in a row. So on position 3, we don't only have an S, we also have, for example, F or, well, Y, C. So we have multiple substitutions that are likely to occur at this position, or, well, occurred in some of the alignments.
Exactly,
And
that screams motif to me. So it screams like, you know,
it is kind of like a motif, you could say. So, I mean, you see how likely are those amino acids to appear in the window. So that's basically, if we go by the column for M, you see how likely is it to appear in the window. Well, it's only likely to appear at one position, but then you have to normalize it by something.
Okay. All right.
And my normalization was, okay, compared to all amino acids that appear somewhere in this window in the alignments. And those are all the positive scores. Does make more sense to you?
Yeah, I'm just wrapping my head around it, but I'm almost.
I mean, for amino acid composition, you do normalize by something, most likely by length of the protein.
I forgot that you had a sliding window.
Ah, okay.
Yeah.
Anybody else has a question related to this method?
So basically it's just like, how likely is it that it's random at that position? So that it appears by random mutation, or that it appears because it has an importance?
So a positive score basically means that it appears because it's somehow conserved. So it should be important. So, okay, I thought PSSM scores were known. So it's basically a positive number in the PSSM means it's more likely than just by random chance, based on the background distribution in the database, to appear at this position. And negative means less likely. So the positive scores are probably somehow conserved. The negative ones are, well, conserved against, basically. And zeros are most likely random. Okay. So our total feature set for this random forest is then, for the global amino acid composition, 40 features, because we have 20 amino acids, and we have the composition for the positive and the negative scores. That's why we have twice that many. We have the binned protein length, which is one feature. We have the just raw PSSM score for every of the 19 positions. In this case, it's 21 by 19, not just 20 by 19, because we introduce one amino acid that just signifies start of the sequence and end of the sequence. Because if we want to predict the first residue in the sequence, everything to the left in the window doesn't exist. So either we could say that's a missing feature, or we can make a feature up. In this case, we have this 21st amino acid that we're using.
But I do, and this actually ties in to my last question, and that was actually something that I was a little bit puzzled by. If we have like a negative feature set and a positive feature set, so those help us to discriminate between the two cases, right? We want to know if either there's a signal peptide or if there is none. Like, that's the problem that we are facing, right?
So the problem that we're facing, that we want to get out, is not just if there is a signal peptide or none, but so we don't have multiple binary, but we have 3 different classes: signal peptides, transmembrane helix, and soluble residue. Is that what you meant?
Okay. Well, because I don't understand why we need to have like the negative cases also, because wouldn't it be sufficient to only have like a positive feature set? And that would tell us if it is like the kind of structure that we are looking for or not?
So yes, most of the information comes from the positive, so from the features based on the positive PSSM scores, but there's also a signal in the negative ones, because it might just be that you have a strong conservation against charged residues, for example.
Oh, okay.
And the rest might be maybe slightly above 0, slightly below or whatever. So the rest is not that conserved in what it is, but the main signal is we don't want to have anything charged at this position.
Okay, I see.
So that's why I also took the negative ones.
I mean, that's basically also zeros. So you have actually 3 groups. You have positive, you have 0, and you have negative.
Yes, but zeros are not that likely to appear in the PSSM, and that's also why I left them out. So I didn't put them into either one, and I didn't consider them as a third category. Okay, so yeah, the PSSM scores that we're using, which is 21 by 19, so window size by this 21 amino acids. The distance to the N and the C terminus, which are 2 additional features. Then the average hydrophobicity for the 9 residue window, the percentage of hydrophobic residues, the percentage of positively and negative charged residues, and the percentage of polar residues, which in total add another 10 features. So the complete feature space is 452 features that this random forest is using. Okay, so now we have our random forest for the initial prediction. We can run a protein through it, and for each residue in the protein we get 3 scores corresponding to a signal peptide, transmembrane, helix, and soluble. But this prediction might look rather, well, noisy, like you've seen in this slide. So we have some outliers in there. And for this, I ran an empirical filter over it. First, I smoothed the scores by running a median filter over it with a window size of 5, which basically just means that I'm taking a window of 5 residues, and the central residue I'm assigning the median value of those 5 residues. Everybody knows what the median is compared to mean or average.
If you like, I don't know, sort 5 values, then it's the third.
Exactly. So basically median means half of the distribution is higher and the other half is lower than this value. I also have an example on the next slide. The other thing is my data set was quite unbalanced regarding the residues. I have many more soluble residues than I have transmembrane or signal peptide residues, and I have more transmembrane residues than I have signal peptides. Which is why I noted an overprediction, and I tried to balance it by adjusting the scores that I get for soluble and transmembrane helix prediction by 185 and by 60 respectively. So if you calculate it back to probabilities, this corresponds to 18.5% and 6%. So basically divided by 1000, and then you have 0.18 and 0.06. which then is the probability. Okay, and after I've run the median filter over it and I adjusted the scores, then I said for each residue, okay, the highest score is the state that I'm assigning to this residue. After that, I filtered out some nonsense, for example, signal peptides of less than 4 residues or transmembrane helices that are less than 7 residues. And after this prediction was done, then I said, okay, yes?
If you have a transmembrane helice predicted and it's below 7 residues, do you assign this area with the surroundings?
Then I'm assigning this transmembrane segment as soluble.
Alright, so definitely soluble.
Yeah. I'm not trying to, for example, say, okay, it might be signal peptide. In this case, I'm just saying, okay, soluble.
Okay. Is the same goes for the signal peptide?
Yes.
Below 4, so you just say soluble.
That's basically the default fallback. And if I have some transmembrane helices left after these last steps, then I make my first classification into soluble protein and transmembrane protein. And well, everything that has a transmembrane helix left is considered a transmembrane protein, everything else is a soluble protein. So here we just have a quick example for this filtering and adjustment step. So first, for example, we're looking at these 5 scores. So as we have a window size of 5, we're looking at the central residue, which is this A, and then, okay, the median of these 5 scores is 600. So we're assigning 600 to this one. That I'm doing for the 3 scores along the whole sequence. After that, I'm adjusting for the overprediction value, so it's quite possible that I also get negative scores, but because I'm just comparing which one is the highest, it doesn't really matter if it's negative or not. And based on the highest score, then I'm making my prediction. So in this case, I would predict a signal peptide. Okay, so now we have the first step, where we just predicted per residue our 3 scores. We had our second step, the filtering step, where we assigned, based on these scores, some kind of annotation to our protein. Now we could be finished, but not quite, because, for example, here, like I said, okay, we suddenly have a transmembrane helix which is 60 residues long, which is probably too long. So in the third step, we try to refine this prediction. For example, try to shift transmembrane helices, make them a little bit longer or a little bit shorter, or such really long segments we might try to split. But first, what are we using for this one? In this case, we're no longer using a random forest, but we're using a neural network. The reason is just because it worked better than a random forest during the cross-validation. So this is one part of the parameter optimization, but I didn't optimize a parameter, but the machine learning tool itself. For this, we went away from a window approach because now we have segments, so transmembrane helices, which don't have a fixed length. They might be 17 residues, they might be 24 residues, whatever. So if we still want to use a fixed length for an input vector for the features, we would have to somehow define a length for this vector, which might be the longest segment we could imagine. But then for shorter ones, we had to leave out some features because we just don't have the length. Or we would have to cut long segments if we have a shorter vector of features. But I tried a different approach. I used just features that are position specific. So I no longer say this is the first residue in the segment, this is the second. Which also meant I lost some information, but it still worked quite well. So what I used as features for this are, once again, the amino acid composition, which isn't dependent on the position in the sequence. I used the average hydrophobicity of the segment. I used the percentage of hydrophobic residues in there, the percentage of charged residues, and also the length of the segment, because that's also a feature that helps if the segment is too short or too long for transmembrane helix, then the neural network should learn that. And the neural network basically is just trained on real segments from transmembrane proteins. So it takes the soluble loops between transmembrane helices and the transmembrane helices as input samples and trains on those to distinguish between real transmembrane helices and non-transmembrane segments. I also permutated some of the segments, maybe by elongating it by 1 residue, shortening it by 1 residue, and still used it as the same class because it's just minor deviation, which is still good enough, but helped to increase the prediction performance because I just had more samples. Okay, so how do we use this neural network? Well, first thing that we try is splitting long helices. So if we have a transmembrane helix which is longer than 35 residues, then we see, okay, can we find a combination of 2 transmembrane helices which are at least 17 residues? So that's basically 2 times 17 plus 1, which is just a small soluble gap between those 2 transmembrane helices. Or if the long helix, for example, was 40 residues, then we would have more combinations because we can make the gap longer or make one of the helices longer. So the number of combinations we can try basically depends on the long transmembrane helix we want to split. And if we find a combination of two shorter transmembrane helices that have a higher average score than the long one, then we take those two. So basically then we decide, okay, it was probably a false prediction where we just merged two transmembrane helices that are closely together. And now we say, okay, in reality those are two separate transmembrane helices. The other thing that we're using the neural network for is to adjust the position of the transmembrane helices. So we shifted maybe one position to the left or to the right or in both directions, or make it a little bit shorter or longer. In total, I tried every combination where I adjusted the endpoints of the transmembrane helix by up to 3 residues. So 3 to the left, 3 to the right on both ends are already enough combinations. If we would make this 10 residues, we would have many combinations which would increase the runtime, and also the probability that we might get one outlier that could completely ruin our prediction. Are those two approaches clear? Does anybody have a question to it? Okay. So after the third step, we now see, okay, we decided to split this transmembrane helix into two shorter ones. As it was 60 residues, we had quite a huge gap we could introduce. This is a little bit shifted, but we are more or less satisfied with the original prediction. So now after the third step, we have our refined prediction for the transmembrane helix. Last step, fourth step, is then just the inside-outside topology. So which of those solvable loops are on the cytosolic or on the extracellular side? Yes?
Oh, no, I think I get it. I was wondering because you introduced, through the split up, you introduced a gap between those two transmembrane helices, which makes sense because you have those 2 points where the decision is not clear, where the two graphs actually attach each other. But I was wondering why the one extreme solvable outburst. No, on the right.
So this.
Yeah, exactly. Why this point is still in the transmembrane helix.
So this is filtered out. So that's why I'm using this median filter, because single peaks are filtered out with the median filter.
So the two left peaks even it out. Sorry.
Yeah.
So the peaks, you believe that the peaks don't carry information or don't carry important information?
Well, some might really be correct. So, I mean, there could be that I have a gap and only one of maybe 4 residues which are in this gap between two transmembrane helices I predict as solvable, but more often than not it's the other way around, and I have a transmembrane helix and suddenly in the middle I have one outlier which I predict to be solvable, and then I would ruin my prediction because I would have 2 transmembrane helices instead of 1. But as I can have also the other way around, that's why I introduced this refinement in the third step.
I might have a stupid question, so please pardon me.
I don't think there are stupid questions.
Is there a significant trend that you could infer the average length of transmembrane helices within one protein is more or less the same? Is there a huge spread of length of transmembrane proteins?
No, so I don't know if the distribution of helix length within one protein is more similar to within all, but I would imagine that the distribution within one is more similar because they span the same membrane. So yeah, but overall there's also not that much of a spread. So the peak most of the time is 21 residues, and then I think most of the data points are within 5 or 6 residues to either side. So the length is already used as part of the segment based.
Yeah.
That's why I used it. Yeah.
I just wanted to add something about these small peaks. I mean, basically, if it's something is a transmembrane helix or not depends mostly on the majority rule. So if you have here very few residues that would be soluble and more like just very short instances in the helix that you would have more likely water. If they're surrounded by others that would most likely be in the membrane, it just evens out and yeah, there's this majority rule, so you really shouldn't just listen to these small outliers if the rest is fine. Doesn't make sense biologically.
Of course, no, but if you look at it, intuitionally, if I would have to manually draw lines, I would also argue that the one where it's ambiguous, I would say, okay, might not be so clear and the signal is inferring, so I would make a cut as well. But afterwards, it still goes up, and as you have the one peak of green within, you could argue that you could also again split the second or the third transmembrane helix into two separate parts with a small loop somewhere.
Yes, you could also argue that maybe you could split this one again, and the algorithm indeed also tries this. So it's always an iteration between splitting, then adjusting position, and then once again splitting.
Using the average score and checking whether it's bigger or not.
Yeah.
I suppose the thing is that we want to have a maximum of accuracy in our predictions, but you can never have 100% accuracy.
Yeah, sure.
So at some point you have to make decisions.
Yeah. Yes.
I'm not sure if I understand this, but does your model account for helices that are membrane but are not transmembrane? So we saw that in one of the previous lectures.
So
cases where they do that.
So basically, what you mean is if I account for re-entrant regions. Yeah. Yes and no. So basically, I'm not predicting them. Mm-hmm. And during the training, I also explicitly consider them as soluble, because they might. If I would consider them as transmembrane, then I might get more false positive transmembrane helices, which in the fourth step would then just screw up my inside-outside topology prediction. So that's why during training, re-entrant regions are at the moment considered as soluble.
Is it actually feasible to predict like integral membrane proteins or membrane-associated proteins?
That's what we're doing. So integral
When I say integral, I mean one like what he said. That's anchors. Yeah.
So basically non-transmembrane but proteins. Well, there are methods that try to predict re-entrant regions, but I'm not too sure how well they perform, because the problem is just that the data set for re-entrant regions is really low.
Well, just simple anchors, GPS.
Yeah, okay. Yeah. So for signal anchors, there might be better prediction performance.
So that method's around. It remains unclear because there's so little data. It remains unclear how well they do.
Yeah. Okay, so now we're coming to the fourth step and the final step for TMSEG, which is the inside-outside topology. Once again, now we're using a random forest. And what I'm doing is, first, based on this prediction, I'm splitting the soluble loops into two sides. And I'm just starting at the first one, and I say, okay, this is side 1. Then after a transmembrane helix, I'm switching the sides of the membrane. So this is side 2, side 1 again, and side 2. And then based off the split, I'm trying to get my features once again. So I have my amino acid composition of those two sides. I have the percentage of positive charged residues on each of those sides, and I have the the percentage shouldn't be in there. The absolute difference of positive charged residues between side 1 and side 2. Do you know why I'm so fixed on positive residues?
Because of the polarity of the lipid bilayer, maybe?
No.
Right. The thing is that it's on the outside of the membrane, but it's on a certain side. I forgot which one, but basically it's also in these which is inside the cytosol and which is outside, so there's a clear preference.
There's a charge gradient between the inside of the cell and the outside of the cell, which is maintained by the cell itself, actually. So.
And also it has something to do with how they are inserted into the membrane, because the translocon which determines which it ends up, which protein ends up, which part of the protein ends up in the membrane. It also is something that can detect that positive charge and say, okay, we're going inside.
Yeah, so that was also already more detailed than I would have expected. But last lecture, I think you briefly also heard about what is called the positive inside rule, where it was determined that for transmembrane proteins, the site that is inside, so most likely cytosolic, has more positively charged residues than the site that is outside, so for example extracellular. And it's not always true, so you can't say, okay, if you go by this rule then you have 100% accuracy, but most of the time it works, and that's why I also used it as a feature. But that explanation was much more detailed.
You can, in fact, by adding charges, you can make the translocon inverted.
I have a lecture on membranes and membrane proteins, and just yesterday I had all that mechanism, which is why I'm reading everything here now in that sense.
Okay. Okay, so basically that's what I'm using as features again. Compared to the other ones, it's not that complex. I'm just using the amino acid composition and then focusing on the positive charge, but it works really well for the prediction. One thing is that I'm not using all the residues in the soluble loops, because I might just have a domain in there. So we could have a transmembrane helix, then we have a domain, and then we have another transmembrane helix in the protein. This domain could maybe be 300 residues, and the other side of the membrane I have only short loops with a total of 20 residues. Then I could just have, due to the sheer number, more positively charged residues on one side than on the other. And that's why I'm only considering the 15 residues that are next to the transmembrane helices, and to account for maybe an incorrect prediction of the membrane boundary, also 8 residues inside of the predicted transmembrane helix. For this partition into site 1 and site 2, and for the feature generation. And then the random forest just gives me a prediction for the inside-outside topology of the N-terminal residue, and based on this prediction I'm extrapolating it over all the other soluble loops. So if I say the first one is inside, then after each transmembrane helix I'm switching sides. That's also one of the reasons why a incorrectly split transmembrane helix would screw everything up after this point.
You briefly motivated 8, the choice of 8.
So the intuition was it's roughly half of the membrane. So even if I... Well, it's a little bit less than, for example, half of 17, which are one of the... You mean because I have 2? Ah, okay. No, that wasn't really the logic, but it was also part of the parameter optimization. So shorter or longer ones didn't work that well. Yes?
Is there a reason why not to also predict the topology of the C-terminus in order to have a validation of some kind? Because if you extrapolate from the N-terminus and you have a mismatch to a C-terminus, you have like a...
But then I would have to somehow use different features, because if both are using the same features, then I guess I'm...
Sure, but as you said,
yes.
There's a non...
Yeah, but then if I... Okay, but if I get contradictory predictions... How do I solve it? Yeah, so that's one of the small things that I will come to later on.
Okay.
But, yeah. Okay, and the one exception to this is just if I have already predicted a signal peptide, I'm no longer doing the topology prediction, but I say what comes after the signal peptide is outside, and from there on I'm extrapolating. Okay, so now TMSEG is basically finished. We have our 4 steps of the prediction, and we have our final prediction for the protein. So first we had our scores for a rough per residue prediction, then we filtered it to remove those outliers, then we adjusted it with the neural network to get a refined prediction, and then finally we try to predict the topology or the orientation inside the membrane. So what's the next step? Of course, we want to know how good are we compared to other methods, or how good are we in general. One thing is that per residue scores aren't really that good for transmembrane helix prediction. One of the reasons are those outliers, because if I have a transmembrane helix of 21 residues, 20 of those I predict correctly, but one in the middle I predict incorrectly, I would have a quite good per residue score, but the overall prediction wouldn't make too much sense, because then I have 2 short transmembrane helices. Which is why we focused on scores that are based on the transmembrane segments. And for those we also have then whole protein scores, the QOK and the QTOP, which I will explain in the following slides. First, you can also just do rough recall and precision for the segments. So basically how many of the correctly predicted transmembrane helices are really transmembrane helices, and how many of the observed transmembrane helices do you predict. And QOK is a measure for my test set. So if I have 100 transmembrane proteins in there, and for 60 of those I predict every single transmembrane helix correctly. So I basically have recall and precision 100% for this protein. Then I also counted for the QOK. So if I have a QOK of 60% out of 100 transmembrane proteins, for 60 of those I'm predicting every single transmembrane helix correctly. What is a correct prediction? Well, that's the other thing. I mean, for segments, you have multiple ways. You could say exact overlap, which is, well, not really the case for most predictions. So then you would have an abysmal performance. But we also used a more strict criteria than what was previously used, because previously you often had maybe just an overlap of 3 or 5 residues. And if you have 2 transmembrane helices of 21 residues, you have an overlap of only 3. Would you still consider this a correct prediction? Probably not. At least a biologist wouldn't.
Historically, yes.
But biologists too?
Whatever you call biologists. Most people in the past who ever measured did.
Okay.
That defines probably yes. But you can well say this is not my intuition.
Yeah, so I guess for me it wouldn't be correct enough. And so the criteria that Ada Kloppmann, Jonas Reeb, and I then tried in 2 different publications was that we're saying, okay, first of all, the endpoints of the predicted and the observed helix, so the start here and the end, they may not deviate by more than 5 residues. 5 residues each, so if we deviate by 3 on the start and by 3 on the end, it's still a correct prediction. And the other thing is that the overlap between the 2 segments has to be at least 50% of the longer one. That's just introduced for very short segments, because then you could get the case where a deviation of 5 residues at each position is actually more different than if you go by 50% overlap. And yeah, then we consider the correct prediction. The other thing for the topology, well, I say the topology for one is correct if the N-terminal topology is correctly predicted. So this measurement would be independent of correct prediction of the transmembrane helices. But we also have the QTOP, which is basically just the QOK with the added criteria that also the inside-outside topology of the protein has to be correct. Do you all understand these measurements, or something unclear? Okay. So how good are we? Well, at least
sorry, I was going to ask the same question. So, do you have the QOK and QTop criteria for the other prediction methods?
Yes.
No, I mean how they define QOK and QTop.
Oh, so
the same 5 residue shift allowance and 50% overlap.
No, so these methods, they all use just a simple overlap, but these values are not based on what was in their publications, but we evaluated them on our test set. So these measurements are on the same proteins and with the same criteria. And well, on average, TMSEG performs best in QOK and QTOP. Though the problem is the error. I only have a test set of 41 proteins, because I only had those 166 to begin with, which is why we have quite a high error margin, and with maybe the exception of polyphobias on this one, we can't say that it's significantly better, because it's still within one error. For the QTOP, at least there we can say, okay, it outperforms polyphobias, but the other ones you could argue are already within one error, and so we can say there's a trend that we're better, but we can't significantly say it. For a per protein evaluation, we simply have the false positive rate. So how many of the soluble proteins do we predict as transmembrane proteins? We have the sensitivity, so how many of the transmembrane proteins do we predict as transmembrane? And those we not only compare to those state-of-the-art methods, but we also have a simple baseline predictor, which is just sliding window of 21 residues. It makes the average hydrophobicity of those 21 residues, and if it finds a window with average hydrophobicity above a certain threshold, it says, okay, these 21 residues are transmembrane helix, finished, basically. And inside-outside topology is just absolute higher number of positive charge. So the simplest way you could get. And here at least we can see that on the false positive rate, TMSEG outperforms all the other methods significantly, because even considering the error, we're basically outside. For the sensitivity, well, we have the problem that every method basically predicts all the transmembrane proteins as transmembrane. So they're even. But the real difference is on the false positive rates, and here you can see that MEMSAT3 and MEMSAT SVM have quite high false positive rates, and well, the baseline predictor has the highest, but as I said, it's just a simple method. If we look at the topology, so like I said, this is independent of correct helix placement. We can see that TMSEG and MEMSAT3 are the best methods. This is also the reason why I included MEMSAT3 in this evaluation and not just the newer MEMSAT SVM, because in previous evaluation papers I've seen that MEMSAT3 performs much better in the topology prediction than MEMSAT SVM, and as I'm predicting both TM helix placement and topology, I wanted to include it to try to compare at least to the statistic. And well, I managed to get the same performance as MEMSAT3 on the topology and better performance on the transmembrane proteins. And if we want to put it in numbers, so if we are taking those statistics, applying it to the human protein, we could estimate that about 560 proteins are misclassified out of those 20,000 from human. And polyphobias, the second best one, well, it has about 50 or well 35% more misclassifications. So while it's more, it's not that extreme, but here you can really see that the high false positive rates from the MEMSAT methods make a huge difference. And that's just because we estimated 24% of the human proteome are transmembrane protein. So we have 76% of those proteins which are soluble. And if you have a high false positive rate, then you have really many misclassification errors. Okay, the other thing that I tried, because working on TMSEG, improving it, writing the paper until it was finally published, took almost 3 years. I just thought, okay, let's look at new transmembrane proteins that we have since then and evaluate the methods on this new data set, because that's something that neither of the methods have seen before. Well, unfortunately, it turns out I can only get 12 transmembrane proteins that are not similar to the ones that I had before in all of those 3 years. There were more than 12 that were added after this date, but many of them were identical or at least quite similar to the ones that I already trained on, so I couldn't use those. We evaluated on those 12. Well, we predicted 10 of them completely correct. So we had the placement of the transmembrane helices and the topology correct, but with 12 proteins in the data set, we can't really say anything about statistic significance, because one protein makes a difference of almost 9 percentage points. But the other thing about TMSEG is, as it's modular, we have those 4 different steps. Only the first and the second step depend on each other, because for the second step, for the filtering, we need some kind of scores, and from other methods you don't really get those scores. You get the final prediction, maybe some confidence score, but most of the time you don't really get for all classes a probability, so we can't really use those steps. But the third, so the neural network for the prediction adjustment, and the fourth step for the topology prediction, you can basically apply to any annotation of transmembrane helices that you have for the protein. And so we tried if we can improve other methods if we apply the neural network and the random forest after, for example, Polyphobius made a prediction. Well, what we've seen here is that for the QOK, we can increase MEMSAT3, which was one of the not so good of methods. But for the other, taking into account the error, on average we're not improving it and we're not making it worse. But for the topology, we can see that with the exception of MEMSAT SVM, we can improve the QTOP for all of the other methods. So even if we take their annotations, we can still, with just those 2 simple steps, we can try to improve the prediction. Now we're coming to something that we could do in the future. Like we previously mentioned, those reentrant regions. At the moment when I made the method, the problem was we just have too little data. I don't have a number what we have today. Maybe we could try to work on it and also predict reentrant regions as a new class. Or another thing that we could try is if we have a transmembrane helix that is predicted with not too high probability, we could, for example, see, okay, if we leave out this transmembrane helix and then do the topology prediction, do we get a higher confidence score? So maybe before we had a prediction probability of 60% to be inside. If we leave it out, we suddenly raise it to 90%. Then we could argue, okay, this transmembrane helix might in fact be a false prediction and it might be a reentrant region. And if we leave it out, we have a higher confidence for the topology prediction and just label it reentrant or maybe solvable. That's something you could try. So, and the last thing is just about dataset size. I already mentioned that I had too few transmembrane proteins to say that I'm significantly better than the other methods because the error bar was high. The other thing is just in terms of machine learning. Usually you want to have at least 1 order of magnitude more samples than you have features or free parameters. Well, in my case, I not always could fulfill this criteria. So basically for the first step, for the random forest, we have our 452 features. And sample size, I have 2,800 residues which are signal peptides. So that's not quite 10 times, but it's roughly a magnitude more.
Wait a minute. 552 is not the feature size, or the free sample, the free.
For the random forest.
Yeah.
What is it?
How many trees do you have?
100.
100 times 9. It's 900.
But due to randomly sampling, you sometimes might end with more or less. I would argue it's more the features.
152. It's larger.
But increasing the number of trees doesn't always really make an overtraining.
You could
So
have tried to cap it, but in principle the number of free parameters is larger. Those are free parameters.
yeah, in the case of random forest, I would argue it's not really that much correlating to the trees.
The number of free parameters. It's the number of things you fit.
Yes. No, because we're still fitting to the features. We don't suddenly have 900 different features. We're still using the original 452 features. We might just use a subset, but...
So in the neural network, by the same token, you argue that you have 47, not 47, 7 times 25.
No, because there we have combinations of the features
Still.
due to the hidden units.
The other one too.
Well, I would...
decisions.
I would argue that it's more related to the number of features in the random forest, but okay. Well, but at least, so here you can see there the sample size. So this is signal peptide residues, transmembrane residues, and soluble residues. There we have quite a few enough samples, so to speak. Even if we say that we have 900 free parameters. For the neural network, it's a little bit different. There we have 47 features, 25 hidden units, so roughly 1000 free parameters and only roughly 2 times the sample size. So there we're slowly getting into the problem of too few samples to really get a good generalization, but it still worked. And for the last one, well, we have more features than we had sample sizes, or basically we have 40 proteins with where we have the inside topology on the N-terminus, 40 where we have the outside topology. So those are our 40 samples to our 86 features. But in this case, we still at least didn't run into the problem of overfitting. So the random forest is really good at not overfitting because of the randomly sampling of the features. So we are not that much dependent on it. And like we've seen on the improvement here or on the overall prediction, we're, well, outperforming most of the rest. So, and because this is on an independent data set, it can't be overtraining in this sense.
Is there anything systematic about the things where you misclassify?
So you mean, for example, on the protein level?
Yeah.
Well, there are some cases where I'm still picking up a signal peptide and predicting as a transmembrane protein, but those are only a subset of my classification. So it's not that I can clearly say, okay, I have a problem with signal peptides because most of my misclassifications are signal peptides. Some of my misclassifications are transmembrane helices, which are somewhere in the middle of the protein. So might maybe be the hydrophobic core of some domain. And the other way around, sometimes for single span transmembrane proteins, so transmembrane proteins with only one helix, I might just miss the signal and predict it as soluble. But I couldn't find a clear picture on what my problem is, so it's pretty balanced in this sense. Okay, so last thing is TMSEG. Where can you get it if you want to use it? It's free to use, and you can either get it from our Debian repository at RostLab, you can get it from GitHub, or you can use it as part of Predict Protein, either on the service or on our web service. Okay. Thank you for your attention.